There is a world out there filled with wonders and beauty. The shining stars, the colorful birds, the deep blue oceans, and of course, us handsome people. Unfortunately, not all of us can see this, especially those suffering from blindness. There are the main, especially from blindness, which could be due to age-related degeneration or genetic disorders. So the main reason for retinal blindness is the death of light-sensing cells in the eye called as photoreceptors. For many decades, scientists have been working on therapy to cure blindness, and one such novel technique is optogenetics. So optogenetics involves inserting DNA of light-sensitive molecules into biological cells to control their activity. Using this technique, we can insert ion channels into neurons and influence their firing rate. So this means that we can insert light-sensitive ion channel like channel rhodopsin into the retina of a blind patient as a replacement for the dead photoreceptors, and voila, they can see again. But is that it? No. Now we have to ensure that these patients have vision as normal and as good as possible. So my PhD project focuses on understanding the differences between normal and optogenetically modified retina and to figure out what the best way to stimulate them in order to get as normal vision as possible. To do this, we use mice as model organisms which express channel rhodopsin in the retinal ganglion cells, the output cells of the retina, and we record the responses to natural scenery before and after activating the channel rhodopsin. We saw that the responses in these two conditions had different properties with varying response curves. This was also specific to the types of cells which were present and active in the retina. So now, in order to bridge the gap and make the responses similar, we modified the natural scenery to make it more suitable for optogenetic stimulation and presented it to the retina. And now we see that such an optogenetically modified retina also responds similarly to the normal retina. Thus, with our results, we are one step closer to providing better and more accurate optogenetic therapy for blind patients. With this, we can help them have more shining stars, more colorful birds, deep blue oceans, and of course, us handsome people in their world.